So if you were like us wondering what just drifted by to the right of the right-hand laser, it was a plastic cup. But maybe more interesting on the left is one of those giant sea pens that we collected one of earlier in the day. It seems like a single polyp octocoral or sea pen, and apparently maybe new to science.
Jeff, have we officially started our transect?
We have officially started our transect. Oh, and we have something very interesting coming up along here. Can't yet see what it is.
It might be a sponge.
It's very bright, so yeah, it's probably a sponge. Yep, that's what it looks like.
Yeah, it might be one of those.
Sponge on a stalk.
Yeah, like a Hylanema sponge.
Yeah.
So what were you saying about Craig's research before we started the setting up for the transect?
So Craig over the years has studied whale falls, and it turns out that there are really fascinating ecosystem. These whale falls, they have very large bones, and the bones in whales are heavily invested in fat. And after the carcass is eaten by things like those amphipods that we saw in the baited trap, and sleeper sharks and other things, these are usually experiments done at much shallower depths than we're at now. But what happens is those bones, that fat, starts to get used by bacteria, and it creates a situation where all of that fat's being burned up and without oxygen in the core of the bones, and it creates what's called the reducing environment. And the chemical gradients from the inside of the bone to the outside of the bone can be taken advantage of by interesting animals that have symbioses with bacteria. Things like bone-eating worms called osedax and other animals. And so there's a whole community that has evolved to live on these whale bones. Occasionally you find some of these animals also at cold seeps, which are natural like methane or hydrocarbon seeps along continental margins. But a lot of fauna is actually specialized. We have something big coming up here to the right of the lasers. You guys haven't seen these before. The lasers are about 15 centimeters apart, so that gives you a sense of scale. This is looking very trash-like to me as we approach it.
Yes, I agree. Like a bucket or something.
That is a bucket or a can, or it's a paint can. Yep, consensus in the control van, paint can. So, yep, sorry to say, but humankind's trash is just about everywhere on the globe. Yeah, we're not under a shipping lane or anything like that, but when stuff comes down here, or things still fall down here. It turns out that a lot of plastic debris is probably in these sediments here, microplastics in particular. There's still quite a lot of organisms here, and all things considered, this is really still rather a pristine environment. Very little influence of humans in this system.
User in your channel stopped recording.
So we had a question in here about how much trash have we actually seen in our, this is dive number 4 at this site.
User in your channel started recording.
So in 4 dives at this site, we have seen 2 cans, a plastic cup, another piece of something that looked rubbery, but who knows what it was. I think that's all I can remember. But yeah, it's certainly far from common. But, oh, and we have a fish coming up. Let me see what this is, because it's way more interesting to talk about than trash. I don't know what this guy is. You see this? Oh, it's got a double shadow, and it's a little stalked sponge. Yep.
Oh, you got me going. I was looking for a fish and I couldn't find it.
Yeah, yep. No worries.
Yeah, but we do see trash on almost every dive we do in the deep sea, and so little of the deep ocean floor has been explored. And to note that there's trash almost everywhere we go is pretty sad. I have a random question for you, Jeff.
This is Jesse now. I'm going to find out to get a charging cord. Can you hear me, Megan?
Oh, I can hear you, Jesse.
Okay, you can hear me.
I'll ask you this question then.
Sure.
What's your fish?
What's my favorite deep sea fish? Oof. Let me think. Give me like 30 seconds to really think about that.
Okay.
Okay, I've got it. Are you ready?
Yeah.
Okay. I think it's a dragonfish. They're actually pelagic, so we wouldn't, and they live in the twilight zone, so we definitely wouldn't see them down here. But they have a cool little chin barbel. It's like a little string coming off their chin. If you're familiar with anglerfish, like from Finding Nemo with the bioluminescent lure, these guys have one of those under their chin. They also have these little strips underneath their eyes that can glow red bioluminescence, which is a lot of organisms can't see red in the deep sea, and so it's kind of like an invisible searchlight that this fish runs around with, and can hunt other fish with like an invisible flashlight, basically. If you're near a computer, if you Google deep sea dragonfish, very, very cool looking animals. They're not very large. They're maybe like, I don't know, 8 inches long, I think.
Yeah, they're pretty small.
Yeah, at most, right? I think we have one in the lab that's one of the larger ones. It's about 8 inches long. They can get pretty small.
I really like dragonfish too.
What's your favorite one? Did you already go through that when I was?
Oh no, I just thought it'd be a fun question.
Oh, it is a fun question. There's too many to really pick one, you know.
Oh, I know. I would change my favorite every day, but I think my favorite is Chaunacops coloratus. It's a type of anglerfish that usually sits on the bottom, and they look like little pink koosh balls.
Oh wow! I don't think I've seen one of those.
They're quite adorable. You usually see them on seamounts, actually.
Oh, okay.
So we wouldn't see them here. The depth is a bit too much for them. But in shallower water in Hawaii, we do see them quite often, and have their gill vents behind their pectoral fins, and they can open and close them at will. And so they sort of use them as siphons in order to propel themselves while they swim.
Oh, that's neat.
Mm-hmm. And they're just really cute in a very
Yeah.
ugly sort of way.
You know, you don't hear a lot of deep sea fish described as cute, so that is nice. I think a lot of people are put off by their appearance, but you know, I respect it.
I think they're really cute.
Yeah.
Other people might look at me and be like, "You're a little strange thinking all these deep sea fish are adorable," but they really are.
Oh, it's just a matter of perspective. Do you have a least favorite deep sea fish?
A least favorite?
No.
I don't think I could choose that.
Yeah, no, it'd be cruel. I wouldn't want them to hear us either on the stream. The fish. I'm gonna hurt the fish feelings.
Then they wouldn't come around, and we really need to see
No.
some fish.
Yeah, we do need to see some fish. We saw one earlier, I think. I think it was Ipnops.
Yeah,
way earlier in the dive.
Yes, we did see an Ipnops.
Yeah.
I think we saw a Cuskeel when we first landed as well.
Oh, you saw a Cuskeel? I must have missed that.
It was right when we got to bottom.
Did you have a question for Jeff? You need to switch over to him?
Oh, ask him the fish question. What's his favorite?
Oh, okay. 1 second.
All right. Megan, can you hear me?
Yep, I can hear you, Jeff.
All right. So I understand I'm supposed to tell you my favorite deep sea fish.
That's correct.
That's correct. My favorite deep sea fish is the rat tail. Preferably the abyssal rat tail, but it is an awesome animal. It is a top predator, and it is a cool scavenger. And yeah, and I guess it's just fascinating in many ways. We think they may make migrations that go completely across the ocean. Oh, and there's a little fish there now. Is that a fish coming up just on the left-hand side?
I think it's a sea cucumber. Is that a fish?
No, I think that's a fish. I think that might be a deep sea lizard fish.
Oh, you're right.
Yep, that's Bathysaurus. That's a deep sea lizard fish, which actually bites the tails off of rat tails. That's one of the ways it makes its living. They've done stomach content studies and found just the tail tips of rat tails in their stomachs.
That's really cool.
Yeah, they are. They are quite the ambush predator.
Yeah, every time we've seen them, they just sort of sit really, don't seem to be fazed by the ROV passing by.
Not until you get very close. We have this great video of one actually out here, not very far from here at the Aloha Cabled Observatory. And it settled down, this deep sea lizard fish, in front of a camera tripod that is sitting there and is on most of the day. And so we're filming it, and this is some of Bruce Howe's Aloha Cabled Observatory stuff. And up swims one of those red shrimp that I think a number of you and a number of other people have seen if they've been tuning in. And this shrimp just happens to swim to lizardfish, and the lizardfish just lunges up off the bottom. Keep in mind, its eyes are not very good. It must have just sensed the fish or the shrimp swimming by and just lunges for it, almost gets it. The shrimp is about 3/4 of the size of the lizardfish, and misses, and it just settles down to the seafloor and just waits again. And that's how it makes its living. It's very much like a lizardfish you would see on a coral reef in shallow water. They just wait for very long periods of time, and then make a lunge.
That's really impressive.
I think the most impressive part was just how fast it came up off the bottom. The pace of life in the deep sea is pretty slow, but this thing, we had to slow the video down in order to see it. It came up within a fraction of a second after the shrimp.
I wouldn't have expected them to be that fast.
They can't do it for very long. I imagine if he couldn't have lunged again for at least quite some period of time. Who knows? Okay, on the left we have another small fish. This is probably Ipnops. He's gonna settle down again, but that's got to be an Ipnops. That little fish on the left. You can see him. He's swimming up again now, and you can see at the head it's really reflective, and that's that pair of eyes. No lenses, just light detectors. Cole, how far have we gone in our transect thus far? Okay, we've gone about 200 meters in the transect.
How long were we transecting for?
We transect for about an hour, and it gets us about 600 meters or so across the seafloor. And what will happen with this video is that in the laboratory, someone's going to watch it, and they can pause, rewind, and basically count and identify everything that we see here. So oftentimes we just have a few seconds as the animal passes, but the person who annotates this video footage can spend some time, consult guides, and actually identify a lot more of this stuff to a finer level than we can do just right now in real time. That'll all be used to estimate the community. What fraction of the animals here are sea cucumbers or more suspension feeders like sponges and some of the cnidarians. It'll give us a better picture of what the abyssal community is like, at least for the megafauna. We call the megafauna all those animals living on the surface of the seafloor that you can see and identify in imagery. And then the word we use for animals living within the mud that are larger is macrofauna. And then the very, very little things living in the mud are called myofauna. So Megan, what other abyssal footage have you seen? You've seen this, of course, and what other parts of the world have you seen have looked at abyssal communities in?
There have been other locations in the. I believe the Okeanos Explorer did a dive in the Marianas at these depths, so we did see a couple soft sediment communities there, and we've also done surveys in the Clarion-Clipperton zone, surveying abyssal seafloor communities that live in nodule fields.
So how does that compare to here?
Well, there's a lot of
in these nodule field areas. How does that compare?
There's a lot of similarity from what I've seen of the different types of animals that we're seeing, but the nodules provide a hard substrate.
There's a shrimp.
Really weird.
Sorry, keep going. I just wanted to notice there was a cool shrimp there.
Yeah, it's cool. Shrimps are cool. I like them. So in the nodule fields, the nodules provide a lot of hard substrate. So you can see more organisms that might live on hard substrates, like primnoid corals, which are a type of octocoral. We've seen some anemones living on top of these nodules, and different types of sponges. Oh, here's another one of those giant octocorals that we saw.
Octocorals, big sea pen. Yeah. So
Actually,
these are actually, we've seen a number of these now. These are not altogether uncommon.
Yeah, those are really cool. And so Chris Ma, who's an echinoderm expert, left me a note on Twitter saying that, let me see his note. So when we collected that first one earlier in the dive, he wrote back to us on Twitter and saying that the umbellulas that we've been seeing all over here have branched arms, but that large sea pen does not have branched arms. So it's a pretty unfamiliar morphology for, deep sea expert that knows a lot about the types of animals we might see here. So it could possibly be undescribed. And then also Steve Oskovich wrote back, hadn't seen anything like this one before either. So we might have something new on our hands, which is pretty exciting.
That is. Oh, there's another Ipnops just to the left. There it goes, scooting away. Yeah, I suspect it is new, and it's pretty amazing. It's a very large animal, yet it's undescribed. The sea urchin that we're working on with Rich Moy, Smithsonian here, we collected 2 of them a couple dives ago. That's a new species. This thing is the size of a soccer ball, and a little bit smaller. And it's possible that some of these large sea cucumbers that we're looking at, the thing we're calling benthidites, may in fact be a new species. So it just shows you how little has been done at locales like this, and how much work we have ahead of us.
That's for sure. It really takes a community to start learning more about these types of ecosystems that are so remote. And now with the technology advances that we have with ROV Lukai, we have the chance to be able to go and survey these areas. So Jeff, since you are the PI on this project, why did you choose Station Aloha as your place of study?
So Station Aloha in surface waters has been studied since the late 1980s, and Dave Carl and Roger Lucas, professors at University of Hawaii, initiated Station Aloha way back then, and it was part of an effort to understand how the central gyres of the ocean, which cover most of the ocean surface, how they function, and how they act to transport carbon out of surface waters into very long-term storage pools where it's sort of out of the atmosphere, surface ocean components. And they have built up an absolutely marvelous set of data since that time about the physics and biology, mostly of the surface ocean. And then starting a number of years ago, originally by Fred Dunabeer and then later by Bruce Howe, they installed the Seafloor Observatory at Station Aloha, which has a variety of current meter sensors and a thermistor chain, things for looking at the physics of the boundary layer of water in the seafloor there. We have a camera tripod set up, and we can observe the animals. So there's a lot of context about the ecosystem at Station Aloha. It's a well-studied place, and that makes it very useful when we come in and are trying to do
the sponges are a little bit more white, bright white. So I think you might be right. It was definitely some sort of sea cucumber. It might be that Amperima that we collected on one of our previous trips that has that really long mouth part, so it stands up off the bottom a bit. And they also have really long feet that they're standing on. People often call them sea pigs.
So I was just about to say that Station Aloha is just one of the sites that we're working on. The other site is called Station M. M stands for Monterey Fan, and it's located off the coast of Central California, and it's at 4,000 meters, so similar in depth to Aloha. And that study site has also been studied since the late 1980s, but the studies there actually focused on the abyssal community on the seafloor, and this was a site established by Dr. Ken Smith, who's now at the Monterey Bay Aquarium Research Institute. And he's been looking at how communities change over time and how they change in settles to the seafloor. There's another Ipnops fish on our left-hand side there. And you can see the detritus, if you look at the tracks, it's like tumbleweeds accumulating along fence lines. You know, anytime there's a little track or a depression, the phytodetritus that sinks to the seafloor here accumulates in those little pits and along those little raised ridges and things like that. And that is the larger fluff that sinks from the surface down onto the seafloor. And that's part of what we're studying. We want to know how important that larger stuff is compared to the more microscopic particles that sink a whole lot slower. So Station Aloha, where we're at right now, abyssal, but in the middle of the gyre, very low productivity system. And then Station M, located off California, just about 100 miles offshore, underneath the California upwelling, super productive, lots of detritus coming down every year. So they're very different places, and we want to understand how the food webs work in each of those, how they contrast with one another.
So since I've only really watched video from the seafloor in places that are the Pacific Gyre in these lower productivity, surface productivity areas, and I'm seeing similarities in the communities, do you think, or well, what do you think about the difference between Station M and here? How are they different? How are they similar?
The communities between Station M and Station Aloha are similar in that they're dominated by echinoderms. That means things like urchins and sea cucumbers in particular, brittle stars. They have rat-tail fishes in both locations. Both locations have these big red shrimp, though they're probably different species. So there's a lot of the organisms are similar, but they are different species usually when you get down to it. So the species of sea cucumber we find off California are different than the ones out here. But one of the biggest differences of all is just the total amount of life on the seafloor. And off of California, where it's super productive and there's lots of food raining down onto the seafloor, the distance that we're traveling now, we would already have counted hundreds of sea cucumbers, literally. They're little tiny pink penny agony with little horns sticking up off the top of their head. These long white sea cucumbers with little long antennas sticking up called Anerophonta. Lots of different species, but just lots of them. The abundance is just so much higher than it is here.
And abundance seems to be in association with the amount of food that's being supplied to the seafloor, right?
Yeah, more food, more animals. I think some of my graduate students and I were talking about this just the other night. I think what's very interesting is that while there aren't that many uh, things like sea cucumbers here, the ones that you do find are quite large, and that's in contrast to Station M over off California, where there's lots of them, but actually most of them aren't any larger than maybe 7 or 8 centimeters in size. Here we find some sea cucumbers that are, easily 50 centimeters in size. They're over 10 times the size, and I'm not really sure what's driving that, but definitely another difference that we have observed.
What advantage does size bring to these animals?
Well, that's the interesting part of all of this, is that generally when food supplies are low, animals are smaller because they don't have to gather as much food to make a living and reproduce. So finding sea cucumbers that are 2 feet long in a very unproductive place like here is a bit surprising. I think perhaps the advantages are that they have a larger gut with which to process over a longer period of time the mud that goes through it. And perhaps they have more of a body wall to store energy when they do gather it. And so they might be able to weather a little bit more variability in how much food they can gather. But I'm not certain. Overall it kind of goes against the general theory that when food is low, you get smaller animals. There's plenty of small animals here. They're just all in the mud. But the sea cucumbers are a definite exception. They just seem to be huge here.
Yeah, that's for sure. The ones we've put in our box today barely fit.
Yeah, I think one of them, yeah, the feet long.
That's just so impressive. You don't really see cucumbers that size just on shallow coral reefs.
No, probably not in total body size. Certainly around Hawaii, most of the sea cucumbers on the reef, they're maybe a foot long. There's a couple of exceptions. There's a species in Kaneohe Bay that's this long worm-like thing that's very skinny and gets a few feet long. But most of the reef sea cucumbers are only about maybe a foot or so.
Oh, yeah.
Megan, we got something coming up on the left. Can you see what that is? That dark thing?
Oh, I think it might be an Hypniastes. It's like a purple sea cucumber.
It looks very sea cucumber-ish. Yep. Probably, yeah. That looks like a Hypniastes, the flying sea cucumber, but it was sitting on the seafloor just there.
But always ready to take off at a moment's notice. I always see them with their little flying wings all extended and ready to go. Do sea cucumbers have predators?
Yeah, there are sea cucumbers certainly have predators. In shallow reefs, there's some. Out on the they're very available to predators, and so most of them, we know this in shallow water at least, most of them have evolved a series of compounds in their tissues that are either distasteful or downright toxic, and so they deter predators simply because they taste awful. They taste awful, they're poisonous, and so they don't get eaten. We suspect the same thing is probably occurring in the deep sea, but we don't know that for sure. We haven't actually tested it. I can tell you that those ratfish that I like a whole lot, they eat just about everything in the ecosystem, but not sea cucumbers. So there's got to be a reason why. It's a little red shrimp or mysid there.
You know what I haven't seen a lot compared to the CCZ? Xenophyophores. I feel like we saw so many xenophyophores in the CCZ, and there are only just a few of them. They're pretty sparse at Station Aloha.
Yeah, that is kind of interesting, but you're right. There just aren't very many of them here. Yeah, I don't know why that is. Xenophyophores tend to live in most deep sea environments, and we've seen a few, but I'm not really sure why certain areas have so many more than others. If it were food, certainly the seas have a little more food than here, but not by a lot. So maybe it has something to do with different species being able to attach to and live on top of nodules, whereas here they would have to develop some sort of anchoring system in the sediments. Maybe there are just fewer species or less capable of doing that.
Well, the dives that we did in the CCZ that had very few nodules, I felt had some of the most, well, large xenophyophores. Whereas the ones with a lot of nodules, there were some nodules that had xenos on them, but the density seemed a lot higher in the areas that didn't have nodules.
Well, that means that the substrate hypothesis is probably shot, and it must have something to do with food supply, or competition for that food supply. So we don't know what other organisms here might compete with or prey on xenophyophores, and maybe what you're really seeing is a difference in abundance of those organisms rather than the xenos themselves.
That's a good
But
theory.
we don't know enough about the system to say. It's just an idea.
Yeah, sometimes you just have to speculate, and that's how you can come up with ideas to base your studies around. What made you think of the idea for this study on the Foodweb?
So the idea for this study, it's sort of a study we had done earlier up higher in the water column in a zone called the mesopelagic or the twilight zone. These are depths between about 200 and maybe 1,000 meters or so, where animals can still see sunlight, but there's not enough for photosynthesis, for phytoplankton to survive. And we've been working in this system, and we had noticed that their isotope composition changed a lot with depth, and we started to tease this apart, and we realized that really what was happening is that you had large, more marine snow type particles, and their isotope values basically looked quite similar to what you see in surface waters, the organisms that are responsible for creating that marine snow. And it sinks down into the water column. But there were all finer particles. We're talking things that are, between 1 and maybe 5 or 10 microns in size. Some of them are bacterial sized. They're not very big. And we had looked at the isotope values of those, and they were considerably higher for a particular type of isotopes we find in amino acids. And this distinction in these particle types, we thought that was interesting. But then what was really cool is that when we looked at some deep water fishes that lived at depths of 1,000 to 1,500 meters, they didn't look like the marine snow. And the marine snow is what everyone was catching in sediment traps and people thought was the main food supply getting into the mesopelagic environment. But these deep water fishes didn't look like that. They looked like the little tiny particles in their isotopes. And so that suggested they were maybe not eating the particles directly. They were fish after all. They don't eat particles, one to plankton and other organisms that were eating that stuff. And the interesting part of this was that scientists had been looking for alternative food sources to the mesopelagic, this twilight zone realm, for a little while. When we measure the sinking detritus that goes into it, there's never enough to support all the energy needs of the organisms that live there. And so we think that part of those energy needs, maybe not all of it, but part of those missing energy needs are these really tiny particles. So we were wondering whether this difference in the composition and importance of these large versus small particles continued deeper in the water column. And if you look at these systems that we're looking at right now, it turns out that when scientists measure the detritus sinking into them with these sediment traps, that there's not enough food down here either. Something is missing in this environment too. The traps probably don't catch all the particles, particularly the small ones, terribly well. So our thought was using in situ filter pumps. Those are not on the ROV. They're deployed from the ship on a wire. And then we can get the isotopes of those small and large particles. And if we're right, and the small particles are really important to the animals living on the seafloor here, then they will have isotopes that look like the small particles we filtered up in the water column a few hundred meters. So basically, we came to these ideas by starting much higher up in the water column and just sort of down here to the abyss.
User in your channel stopped recording. User in your channel started recording.
It's interesting. We can see in our downward-looking camera, we're coming up upon what looks like a trench. You can just see it on the edge of the light field now. It's just like a long trough or low spot. We are wondering what might have made this. So here it is. We've got a little bit of a trench. Uh-oh. Got a little low here. So folks here are wondering if that was actually an old cable burial location where that might have been a trans-Pacific telephone or internet-style cable. We have many of them that run from the U.S. mainland and other points into Hawaii. Okay, we've got about 30 seconds and we're coming up on the end of the transect.
Oh, really?
Okay, Cole, we're calling this end of transect. What was the length? How many meters did we transit? About 700 meters. Okay, great. Okay. Now, we've still got a little bit of momentum from the TMS, but what we'll try to do next is to collect some more animals.
Jeff, I was wondering, who is sponsoring our research here today?
Yeah, so the research is sponsored by the National Science Foundation, and they've funded, there's 4 of us that are co-PIs. Brian Pope and myself, we're both here at C, and Claudia Benitez Nelson at University of South Carolina, and Craig Smith, who's also at UH with us. This outreach is being sponsored by Mike Jackson, who is a charitable donor to the university, and we are very thankful for his support in helping us to provide this outreach. There's also part of this outreach is being done by Kanisa Duncan, who's a faculty member at UH, and Bridget Conter Smith, who's also a faculty member at UH, and Bridget runs a program of REU students, research experience for undergraduates students in the summer, and a high school program as well. And so some of the students from her classes are tuning into our broadcasts. So But that's outreach is Mike Jackson and also some additional support from the National Science Foundation. So now that we're slowing down, I think what we're going to try to do is get this bait trap. You can see the amphipod swimming around in there. We're going to try to stow it a little more securely in the basket, which will free up our arm and enable us to collect some more organisms.
and how we've been dealing with that while we're trying to do our work. So right now, we're actually doing this telepresence to you live, very socially distanced. I am currently in Lab 1, which is on the complete opposite side. All taking precautions, wearing our few people in the same space at all times. As work environment on the ship. Everybody needs having to stay apart while still getting the same work done. And but we have quarantined ourselves for as
with juggling objects in the basket before we start doing a few more collections.
sounds good.
Okay, so we were just discussing strategy. cores. We've got 7 more of them to take. And after we're done with the sediment cores, the remaining time, we've got about an hour and a half left on the seafloor. Cores will take about 45 minutes. And then we'll spend the rest of the time collecting animals. And then that'll be the dive. So low currents today continue to plague us. And low current that we stir up every time we move the vehicle doesn't settle. And so that's part of what we've been wrestling with today. The mud on the abyssal seafloor and almost all parts of the ocean is very, very fine clay. And once it's knocked back up into the water column, it takes a long time to settle or drift away.